Hi, my name is Keisha, and I will be talking to you about glucose 6-phosphate dehydrogenase and G6PD deficiency. Glucose 6-phosphate dehydrogenase, I will be covering what is G6PD, what does it look like, the diagnostic procedures, the malaria research project, management and treatment, and my gene tree. So what is glucose 6-phosphate? It is the enzyme that helps the body turn glucose into energy. It's responsible for the first step in the chemical pathway that converts glucose into ribose 5-phosphate. And I have the chart over here. It protects the red blood cells from potentially harmful byproducts that can accumulate when a person is taking certain medications or when they're fighting an infection. Clearly, me. So what is the deficiency? There's 140 mutations identified of the gene. These are changes in the amino acids that make up the glucose 6-phosphate enzyme. And these changes disrupt the normal structure and function of the enzyme and reduce the amount of enzymes in the cells. Red blood cells are unable to protect themselves when this is diminished, and there's damaging effects on them from reactive oxygen. The damaged cells are likely to rupture and break down prematurely. Excuse me. And undergo what is called hemolysis. Hemolysis can be increased by certain antibiotics, infections, and the consumption of fava beans, and G6PD deficiency is also called favism. And in my little chart over here, I have little red blood cells and the infection like and then I have medication, and it's normal antibiotics, and then fava beans. They're so mean. So how do you get G6PD? Well, I'm going to tell you a little bit about it because I have it. I am affected by G6PD, and I made my gene chart right here. My son and my husband and my son's father. Husband does, and it's passed from mother to son and father to daughter. So because I am affected, that means one of my parents, probably my father, passed it down to me. Thanks, Dad. Appreciate it. Most likely in males of African, Mediterranean, Asian, and Middle Eastern descent. Because it passes to their daughters, and I'm also affected, I'm pretty sure Kaylin probably has it, and we have to get her tested for it. G6PD diagnosis. So, this guy, Ernest Butler, in the 1960s, developed the fluorescence spot test. He was the commissioned second lieutenant in the Army, and he was assigned to work with the malaria research project. He noted that the cells had more Heinz bodies, which are these little spots. The blood cells, when treated with certain chemicals, mainly the primaquine, the malaria drug. So to diagnose, you get your blood drawn at your doctor's office or in a lab, and they put NADP in it and mix it together, and then they put it on spotted filter paper. They put it in an incubator for 5 minutes and 10 minutes, and once it's dry, they put it in under UV light, and once it is under the UV light, they can look at it and see that if there's NADPH, which is the fluorescent, if it shows fluorescent, that means NADPH is there, and G6PD is present. If there's a lack of fluorescence, then that means this blood sample is deficient. So what does it look like? Well, because in G6PD deficiency, your red blood cells destroy themselves and they're removed from your bloodstream, that leaves you with low amount of red blood cells, leading to anemia. So if you're anemic, you can be fatigued, you can experience dizziness, paleness, coldness, yellowing of the eyes and skin, known as jaundice, shortness of breath, muscular weakness, palpitations, rapid heart rate. And in severe cases, you can have chest pain and a heart attack, but and your spleen can be enlarged. So because the spleen is the filter, and that's where all the red blood cells go when they're destroyed, it can become enlarged because your red blood cells are going at a rapid rate. So the malaria research project was done in the Army on African American males who developed acute hemolytic anemia after undergoing therapy of primaquine, which is the drug they give us for malaria when we're deployed to the Middle East, Africa, Asia, and it showed that they were getting very sick, and so they decided to do a study, and they found that those red blood cells in the blood samples were normally 62 days life cycle, were 13 days life cycle in G6PD deficient males, African American males. And it was causing hemolysis, and they were getting tired faster. And it was after they were exposed to the malaria drug. And in the military, it's mandatory that you take the malaria drug up until they decided to do this project. And so 2 to 4 days after initiating malaria therapy, which is with the primaquine, the hemoglobin typically falls 3 to 4 grams per deciliter. But once you take that reagent, once you take that away, the primaquine, and it gets out of their system, their body normally returns to normal. Fun fact, the malaria parasite cannot attack in an environment with premature red blood cells. So again, thanks, Dad. So the management and treatment: avoid the oxidative stressors, anti-malarials, sulfonamides, aspirin, and some NSAIDs like Tylenol, and fava beans. And they said something about peanut butter or peanuts and blueberries. And the treatment is remove the hemolytic crisis, the cause of the crisis, and some in severe cases of blood transfusion. And this is and they didn't do any testing on anybody other than primates. So these are all the primates. And this is me and my baby Kaylin. And thank you guys for listening to my spiel on G6PD Deficiency.